Hello everyone. In this class, we'll try to learn about synaptic potential and different properties of synapses in detail. Synaptic potential is a longer duration than action potential. When it is excitatory, it can cause repeated firing of initial segment of postsynaptic neuron. Synaptic potentials are two types: excitatory postsynaptic potential, which are called as EPSP, and inhibitory postsynaptic potentials, which are called as IPSPs. So this is the diagram showing EPSP and IPSP, where the membrane potential in the postsynaptic membrane changes from -70 to -60, that it becomes less negative, and during EPSP the membrane potential in the postsynaptic membrane, it becomes more negative. Excitatory postsynaptic potential. If the potential changes in the postsynaptic membrane during synaptic transmission is towards depolarization, the potential is called as EPSP. For example, if initially RMP was -70 millivolts, which becomes -60 millivolts during synaptic transmission, then it is EPSP. The latency of EPSP is about 0.5 milliseconds. EPSP reaches its peak at 11.5 milliseconds after the entry of afferent impulses into spinal cord, which then declines exponentially. A single EPSP leads to a change in 0.5 to 2 millivolts only, though the EPSP produced at one synaptic knob is small. The EPSP at many synaptic knobs summate. EPSP of optimum magnitude leads to excitation of the postsynaptic neuron. During this potential, the excitability of the postsynaptic neuron increases to other stimuli, and therefore the potential is called excitatory postsynaptic potential. EPSPs are produced by the neurotransmitters like acetylcholine, noradrenaline. The EPSP is caused by opening of the sodium or calcium ion channels in the postsynaptic membrane that results in influx of sodium or calcium and causes depolarization. EPSP is also caused by closure of potassium ions. Slow IPSP has been observed in autonomic ganglia and in cortical neurons. They have latency of around 100 to 500 milliseconds, and they persist for several seconds. They are usually due to decreased potassium conductance. Inhibitory postsynaptic potential. During synaptic transmission, if potential of the postsynaptic membrane is carried towards hyperpolarization, then the potential is said to be IPSP. This is because of the hyperpolarization leads to inhibition of postsynaptic neuron. For example, if RMP was -70 millivolts and after transmission it becomes -80, that is more negative, then the potential is IPSP, which is of magnitude of -10 millivolts. Actually, very small change occurs due to the IPSP, that is about 0.5 millivolts. During this potential, the excitability of the postsynaptic neuron decreases to other stimuli, and therefore the potential is called as inhibitory postsynaptic potential. IPSP, like EPSP, is a local response and can be summated. The IPSPs are caused by transmitters like GABA, that is gamma aminobutyric acid glycine. IPSP reaches its peak at 11.5 milliseconds after the application of the stimulus and then decreases exponentially. The IPSP is produced by opening of the chloride channels, that is, chloride enters into the cell along the concentration gradient. Opening of the potassium channels that results in potassium efflux can also produce IPSP. Closure of sodium or calcium channels also can produce IPSP. Slow IPSPs are also observed in autonomic ganglia and cortical neurons. They usually occur due to increased potassium conductance. As it is known, the IPSP is mediated by chloride influx. However, it is in an experimental setup, the chloride influx occurs only when membrane potential is less than minus 70 millivolts. For example, the RMP at minus 50 millivolts. At the membrane potential of minus 70 millivolts, influx or efflux of chloride across the membrane disappears, as the equilibrium potential of chloride is minus 70 millivolts. If the membrane potential becomes more negative, such as at RMP of minus 90 millivolts, chloride efflux takes place, and that makes the interior of the cell positive, and this is called as reversal potential. Coming to genesis of action potential in postsynaptic neuron. The message arriving at postsynaptic neuron usually comes from large number of presynaptic neurons. These neurons can end on any part of the soma and dendrites of the postsynaptic neuron. In central nervous system, many excitatory and inhibitory inputs continuously and simultaneously arrive at the postsynaptic membrane. All the EPSPs or IPSPs produced at various postsynaptic membranes act together and summate electrotonically. The soma of the neuron integrates these potential, and the algebraic sum of these depolarizing and hyperpolarizing potential finally produce the change in the resultant potential. If the resultant potential is depolarizing and the change is about plus 15 millivolts, the firing level is reached that leads to genesis of a propagated spike potential. However, a full phased action potential is generated only at initial segment, as this part of the neuron has the lowest threshold. The action potential propagates into two directions: towards the axon terminal, down the axon, that is called as orthograde propagation, and towards the cell body, that is retrograde propagation. The propagation of action potential into the soma cleans the soma for subsequent renewal of postsynaptic activities. Role of dendrites. Dendrites are the extensions of soma. They increase the surface area for presynaptic knobs to end on them. Thus, more number of dendrites better the integration of excitatory and inhibitory activities. Number and pattern of dendritic spine change during development, as a special type of neural protein called as neurolignin controls the synapse formation. Alteration in the dendritic spine occurs in learning and long-term potentiation. It has been recently observed that the propagated action potential are also initiated in few dendrites. It has also been noted that protein synthesis, which usually occurs in the soma, also occurs in ribosome in the dendritic spine that can alter inputs from glutaminergic neurons. Transmission through electrical synapses. In electrical synapses, pre and postsynaptic membranes come close together to form gap junctions. Gap junctions are low resistance channels through which ions pass easily. Electrical activities of one neuron can pass to other directly through gap junctions. There are sparse electrical synapses in central nervous system. Transmission through electrical synapse differs from that of chemical synapse by the following ways. The EPSP generated in the postsynaptic neurons by electrical transmission has a short latency than EPSP generated in the chemical transmission. In chemical synapse, the transmission is usually unidirectional, whereas the electrical synapse, the transmission is bidirectional. The signal is not magnified in electrical transmission, as magnification or modification of signal occurs in the chemical synapse. Chemical synapses are superior to the electrical synapses in the transmission of impulses. The time taken for impulse to travel in electrical synapse is much less than chemical synapse. Coming to the properties of synapses. The synapses, or rather the synaptic transmissions, exhibit the following properties. Law of forward conduction. Through a synapse, impulse travels in one direction only, that is from the presynaptic to postsynaptic neuron. This is called as law of forward conduction. However, the transmission of impulse is bidirectional in electrical synapses. Synaptic delay. The time required for impulse to be transmitted through the synapse is called as synaptic delay. It is about 0.5 milliseconds. The synaptic delay occurs due to the time spent in entry of calcium into presynaptic knob, release of neurotransmitter into synaptic cleft, and for the action of neurotransmitter on the postsynaptic membrane to produce the postsynaptic potential. The knowledge about synaptic delay helps to find out the number of synapses present in neural pathway, that is whether they are monosynaptic, disynaptic, or polysynaptic reflexes. Law of divergence and convergence. Divergence means one to many projections. This means information from one presynaptic neuron passes to many postsynaptic neurons. Convergence means many to one projection, in which many presynaptic neurons project to a single postsynaptic neuron. Divergence and convergence are very common in central nervous system and form the physiological basis for summation and facilitation at synapses. So this is diagram showing the convergence, that is many axon terminals act on one presynaptic neuron, and this diagram shows the divergence where one single neuron, it is going to give collaterals to many postsynaptic neurons. During synaptic transmission, if the potential of the postsynaptic membrane is carried towards depolarization, it is called as excitatory postsynaptic potential, or if the potential is of postsynaptic membrane is carried towards hyperpolarization, then it is called as inhibitory postsynaptic potential, as we know. There are also synaptic inhibitions. In central nervous system, the neurons are highly interconnected, and therefore any arrival stimulus would lead to an widespread activation of many neurons. Synaptic inhibition prevents this explosive situation by stabilizing the neurons and by preventing unnecessary spread of impulse. There are two different types or different variants of synaptic inhibitions. So it can be direct inhibition or indirect inhibition, or it can be a feedback inhibition or feed forward inhibition. Direct and indirect inhibition. So coming to direct inhibition, when synaptic inhibition occurs through formation of the EPSP on the postsynaptic neuron, it is called as direct inhibition, as the postsynaptic neuron is directly inhibited. Example of the direct inhibition is inhibition of anterior horn cells by descending inhibitory fibers in the spinal cord, or by afferent inputs originating from the Golgi tendon organs, so that influences the anterior horn cells via inhibitory interneuron. In both these situations, there is a release of inhibitory neurotransmitter from presynaptic neurons. Indirect inhibition occurs when the passage of impulses through a synapse is inhibited by another earlier impulse originating from separate neuron terminating on presynaptic ending. This inhibition occurs due to the decreased release of neurotransmitter from the presynaptic nerve terminal, so which is called as presynaptic inhibition. Indirect inhibition could also be due to the effect of previous postsynaptic neuron discharge. For example, the neuron can be refractory to excitation because it is in a refractory period as it has just fired. After polarization, cells are less excitable. In spinal neurons, after hyperpolarization may be prolonged, especially after repeated firing. So this diagram showing direct and indirect inhibition at the synapses. In direct inhibition, the inhibitory presynaptic neuron directly inhibits the postsynaptic neuron by releasing inhibitory neurotransmitter. In indirect inhibition, the presynaptic neuron is not inhibitory, but another inhibitory neuron project on the axon terminal of presynaptic neuron that inhibits the release of neurotransmitter from it. Therefore, transmission is inhibited. For presynaptic inhibition, three mechanisms have been proposed. The activation of the presynaptic receptor facilitates chloride conductance, which decreases the magnitude of action potential reaching the excitatory ending. Thus, calcium entry is decreased and consequently the quantity of neurotransmitter released is reduced. Second, the activation of presynaptic receptors also causes voltage-gated potassium channels to open. The resulting potassium efflux decreases the calcium influx. Third is direct inhibition of neurotransmitter release can occur independent of calcium influx into the excitatory ending. GABA was the first neurotransmitter shown to produce presynaptic inhibition. There are two types of GABA receptors, GABA A, GABA B. GABA acting through its GABA A receptors produces presynaptic inhibition by increasing chloride conductance. Through GABA B receptors, it produces presynaptic inhibition via G proteins that produces increase in potassium conductance. The other neurotransmitters also produce presynaptic inhibition by G protein mediated effect of calcium channels and potassium channels. Feedback and feed-forward inhibitions. Feedback inhibition, Renshaw cell inhibition, which occurs in the spinal cord as example for feedback inhibition. So this is the negative feedback inhibition in which collateral from the spinal motor neuron ends on an inhibitory interneuron, which is known as Renshaw cell, which in turn inhibits the discharge of the same motor neuron. A collateral from Renshaw cell also inhibits neighboring motor neuron, which is called as lateral inhibition. Feed-forward inhibition. The example of feed-forward inhibition is inhibition of Purkinje cell output by parallel fibers originating from granule cells in the cerebellum. Mossy fiber inputs stimulate granule cells, which through its parallel fibers activates Purkinje cells. The output of Purkinje cell is inhibitory as neurotransmitter secreted by Purkinje cell is GABA. Thus, stimulation of mossy fiber parallel fiber pathway finally activates the inhibitory output of Purkinje cells, and this is called as feed-forward inhibition. Okay. So this is a diagram showing negative feedback inhibition by a Renshaw cell. The collateral from the spinal motor neurons end in inhibitory interneuron, so which is called as Renshaw cell, that in turn inhibits the discharge of the same motor neuron and also inhibits neighboring motor neuron, which is called as lateral inhibition. Synaptic facilitation. This is the process by which transmission through a synapse is increased. It is usually caused by increase in the neurotransmitter release from the presynaptic terminal. Therefore, it is also called as presynaptic facilitation. Prolongation of action potential at presynaptic ending helps a voltage-gated calcium channel to remain open for longer duration. This ensures more calcium influx that in turn increases transmitter release from presynaptic ending. Thus, transmission through the synapse is facilitated. The serotonin released at the exoaxonal ending causes presynaptic facilitation by increasing interneural cyclic AMP levels, and this induces phosphorylation of one group of potassium channels that closes the potassium channels. This slows the process of repolarization and prolongs the duration of action potential. Summation literally means to add. Since synaptic potentials are graded, individual potential changes can summate. The phenomenon of summation is seen in neuronal networks involving multiple synapses in which synapses are shared by many neurons. There are two types of summations, temporal summation and spatial summation. In temporal summation, the same input stimulates the postsynaptic neurons repetitively in such a way that The second synaptic potential arrives before the postsynaptic cell recovers from the first synaptic potential. This happens because of the action potentials arrive in rapid succession at the presynaptic terminal, which in turn stimulates the postsynaptic membrane in quick succession so that resultant postsynaptic potential overlap in time. Thus, postsynaptic potential is elevated to the firing level. In spatial summation, instead of repeated stimulation by the same input, two or more separate inputs arrive simultaneously at postsynaptic membrane. In this situation, if the inputs have the same sign, either all are excitatory, all are inhibitory, the postsynaptic response evoked by all inputs become larger than the response that would have occurred in response to their individual application. So this type of summation is called as spatial summation. Thus, temporal summation occurs when single presynaptic knob is stimulated repeatedly, and spatial summation occurs when many synaptic knobs converging on one postsynaptic membrane are stimulated simultaneously. When presynaptic neuron, say fiber A, fires 5 postsynaptic neurons discharge, and when another presynaptic neuron, for say B fiber, separately another 5 postsynaptic neurons discharge. However, as two presynaptic neurons share their discharge zone, simultaneous firing of A and B results activation of 8 postsynaptic neurons instead of 10. So this is called as occlusion. Okay, so in this diagram you can see when the fiber A is fired, so there will be activation of the 5 neurons. Okay, and when the B is fired, okay, so it is also going to activate 5 fibers, postsynaptic neurons. But when they are stimulated together, there will be activation of only 8 postsynaptic membranes because of the overlapping, and this condition is called as occlusion. Then habituation, sensitization, and potentiation. The phenomena of habituation, sensitization, and potentiation are also observed in synaptic transmission. In habituation, the synaptic transmission is decreased, but in sensitization and potentiation, the transmission is increased. Habituation is the gradual decrease in transmitter release in response to repeated transmission of impulses. This is probably caused by prolonged inactivation of calcium channels in the synaptic knob or due to decreased number of vesicles in the presynaptic terminal. Sensitization occurs when transmission is accompanied by painful or unpleasant sensation. The response increases more and more by mechanism of presynaptic facilitation. Sensitization is short-lived and it is explained on the basis of increased calcium entry into the presynaptic terminal. Potentiation is enhanced synaptic transmission for prolonged duration. In post-tetanic potentiation, the duration is shorter in comparison to the other potentiation, which is called as long-term potentiation. The post-tetanic potentiation is due to the increased calcium in the presynaptic knob in response to the arrival of series of stimuli that causes increased transmitter release. The long-term potentiation occurs due to the increased calcium in the postsynaptic neuron due to the opening of NMDA channels, that is by glutamate, along with increase in the intracellular proteins. Synaptic plasticity. There are several ways by which synaptic transmission is modified to suit the need of the body and to learn to react against the particular stimulus in an appropriate manner. These changes are memorized for a variable period by concerned cells and that are involved in the process of learning and memory. This involves a long-term changes in the synapses, which is called as synaptic plasticity. So these are about the synaptic potential and some of the properties of synapses in brief. Thank you.